day everyone. Welcome to the week 8 of the course Biological Data Analysis and Visualization with R. In this week we will be talking about classification in biological data. So, this is a very important problem with a lot of applications in biology as well as medicine, where we want to look at sample data from patients or from other studies, and we want to classify them into different classes. Okay. So, this is something we will be taking over for this week, and we will be talking about different methods that can help us do this data classification, and we will also again do some hands on where we will actually apply these methods to do this classification in actual data sets. So, this is what we are going to talk about in this class, right. So, we will first introduce data classification. I will also talk about what are the applications, and as we go through the presentation as well as through the hands on, you will understand how data classification is helpful and has a lot of applications in biology as well as medicine. And then we will be talking about different classification methods, and in particular we will be talking about 3 classification methods, which I will introduce a bit later, and then we will talk about each of them, and we will do hands on with these methods. So, let us now first discuss what do I mean by data classification. So, when I say data classification, we are looking at sample data, and we want to classify into different classes. Okay. And this classification, this problem has a lot of important real world application. So, I can give you some example. For example, we are looking at, let us say, data from patients and samples where we have to classify them according to whether they fall into normal category or disease category, right. So, this kind of predictive again approach could be very useful for example, the diagnostic purposes. So, as I said, this is something some examples, but you can think of many more such examples. For example, we are looking at samples, some characteristics of this sample, some features, right, whether it is gene expression or whether it is some sort of other associated measures from these samples that have been done, and based on those we want to classify these samples into either disease category or normal category, right. So, as you can see, if we can build a very accurate predictive model, this is something that would be very helpful for diagnostic purposes. Similarly, we also can think of this problem in terms of, let us say, cancer subtypes, right. So, we want to identify different disease subtypes, but sometimes they are not so easy to identify just by looking at them, or you have to perhaps do a lot of complex experimental assays on the samples, and this is something, this could be something that is very time consuming and also maybe resource intensive. So, in those cases, if we have a data classification method that can, from the data that we have, can actually classify disease subtypes into different categories. So, that is something that would be very useful and valuable. We can take the example of cancer subtypes. So, typically we have a lot of gene expression data for different cancer types, and based on this gene expression data we would like to classify them in cancer subtypes. So, this is something that is of interest in general, right. So, classification cancer subtypes means there could be different therapeutic treatments, different prognosis of the cancer progression. So, this kind of applications are will have so this kind of data classification will have a lot of applications in real world. So, this is just very simple examples, but you can think of this in terms of many diseases, many types of data, many kind of applications, not just disease samples, but you can also think of other applications as well. So, with that very brief idea about data classification, now we will talk about the data classification methods, and there are 3 types of methods that we will be discussing in this class in this week. So, we will start with logistic regression. We have talked about linear regression before. So, logistic regression will be a continuation of that, but this is something that we use for classification. We will talk about something called decision trees after logistic regression and how they can help in data classification, but I should also tell you that decision trees can also be used for building regression models. So, when I say regression models means we want to predict values of a response variable. So, that is also possible. So, these are called regression trees. We will again briefly discuss when we discuss decision trees. And then finally, we will be talking about random forests. So, this is an extension of decision trees which is designed to address some of the challenges of decision trees. So, these are the three techniques we will be talking about. What is particularly interesting about decision trees and random forests is that they do not require any linear relationship between the independent variables and the dependent variable, right. So, this is something we have seen in linear regression. We assume a linear relationship between the response variable, the y, right, the dependent variable, and the independent variable, the x1, x2, and so on. So, in this case, decision trees and random forests can actually be more powerful than linear regression. Okay, so with that, let us first discuss logistic regression very briefly, its theory, and then we will take on the hands-on part. So, logistic regression can be classified into mainly two types. So, one is the binomial logistic regression or binary classification, where we are interested in classifying samples or data into two classes. So, here is an example, right. For example, we see a sample data, we have gene expression data, we will like to classify into just cancer versus normal. Okay. So, this is an example of binary classification where we only have two classes. Okay. So, this is one example, but often we will have multiple classes, right. So, it is not always necessary we will have only two classes, we can have multiple classes, like let us say we want to classify normal, then in cancer we also want to say whether it is lung cancer or blood cancer, that kind of idea, right. So, you can think of this like we have multiple classes for classification, and this is something we refer to as multinomial logistic regression. Okay. So, this multinomial logistic regression is used for multi-class classification, and as an example, this is again classification of cancer types and subtypes, this which is not limited to 2, there could be many more. So, in those cases we will be using multinomial logistic regression. So, let us now briefly first understand how logistic regression works, and then we again look into the applications and do the hands-on. So, as we have seen in case of linear regression, right. So, the response variable y is a function of the independent variable x's, right. So, here there is a linear relationship, but it is not to the response variable, it is to something called log p by 1 minus p. Okay. And what is this p? And this p is probability of success and 1 minus p is probability of failure, right. So, when I say probability of success or probability of failure, right. So, this is actually related to binary classification, right. So, one class would be defined as success, another class would be defined as failure, right. That is what we are defining here, and this p is measuring that the sample is probability of that a sample belongs to class 1, right, and 1 minus p is the probability that the sample belongs to class 2. Okay. And this log p by 1 minus p is referred to as log odds ratio. Okay. So, as you can now see the difference with linear regression, here we are assuming the log odds ratio is a linear function of the independent variables, and this is what we represent here, right. So, compared to linear regression, this is a different relationship, but nevertheless we assume linear relationship, okay, between a measure of this log odds ratio and the independent variable. Okay. So, as before we are familiar with these terms. So, x1, x2, x3 these are independent variables, then we have w1, w2, w3 is called weights or coefficients, and b is the bias or error, right. So, x1, x2, x3 are also often referred to as features, right. So, we have used that term. So, this is for binomial logistic regression, where we have binary classification, only two classes. Now, what happens if we have multiple classes, right, multinomial logistic regression, this is also sometimes called softmax regression, and this is the formula. So, this is actually a quite simple formula. So, I will break it down in a moment. So, what we are measuring again is the probability that a sample with values x, right. So, x is a vector, so that is why it is in bold, belongs to class C. Okay. So, that is the probability that we are measuring on the left hand side here. Okay. So, this is probability x c, and this is equal to exponential z x c, right. So, this z x c we will discuss in a moment, and divided by summation j equal to 1 to k exponential z x j. Okay. So, what is this PXC? As I said, this is probability that a sample with value x belongs to the class C, right. So, it can have a set of values for x1, x2 parameters. What is Z is this is actually then W1j x1 plus W2j x2 and so on. So, what we are assuming is that this Z is a linear function of these independent variables or features. K is number of classes and M is number of features or independent variables that we have in the data. Okay. So, then what we are doing is simply we are measuring that probability of a sample belongs to class C is given by this function exponential z x c, which is a linear function again of the independent features, and divided by the sum, right, exponential z x j. So, this is again an independent linear function of the independent variables, and we are taking sum over all classes, right. So, what is actually happening is that we will calculate this probability for all classes, right, for a specific sample, and the probability which has the highest value, right, that is likely to be the classification or the prediction for class of that sample. Okay. So, as you can see, the sum of these all probabilities will be equal to 1. As before, right, Wij, these are the weights and the biases, right. These are the weights and biases that we will estimate from the parameter, from the data that we have. These are parameters of our model. So, we will estimate these from the data. Okay. So, with that introduction, we have talked about, we now understand, right, what are the assumptions and how this logistic regression is modeled, right. Instead of directly assuming y to be a function of independent variables, here we are measuring the probability or the log odds ratio as a function of some sort of function of these independent features or independent variables. Okay. So, with that background, we can now move on to the hands on part, where we will be implementing logistic regression. So, particularly we will be implementing multinomial regression. We will do this on a cancer data set, followed by on gene expression noise data set. So, we have seen these data sets before. So, we will just apply now logistic regression on these data sets and see if we can come up with accurate classification models. Okay. So, this is something that will be interested in the hands on. We will also be talking about, so sometimes what we have seen in case of linear regression, that there could be overfitting, right. So, this is something that is also a problem, could be a problem in logistic regression, and for that we have very similar idea like linear regression. These are called penalized logistic regression. So, this is penalized logistic regression is kind of similar in idea to lasso regression and ridge regression. So, again look into how we can implement penalized logistic regression, and we will also talk about stepwise logistic regression, which we have also talked about in case of linear regression. So, the idea is very similar here. Okay. So, with this, let us now first move into the first part. So, in this class we will demonstrate only the logistic regression on multinomial regression on cancer data set and noise data set. And in the next class, we will be talking about penalized logistic regression and stepwise logistic regression, and we will implement those. So, let us move into the R console for the hands on part. So, here we are for the R console, and okay. So, I hopefully can see the screen with R now, and I will be first showing you the data, and then we will implement the logistic regression. So, the first data we will be working on is the cancer data, and this is something we have seen before earlier when we were doing the principal component analysis. And this is the data, right. So, I will load this data 100 gen species to new, right. So, this is the data that we will have used before for PC and UMAP. But before that, we will need these libraries. So, you probably now know what the function of each of these libraries are, right. So, library reader, tidy models, right. So, these are important, and we also need the library dplyr. And we will also load this library glmnet. So, this is again you have seen, right, GLMNet generalized linear models. So, we have used that for example, lasso regression, etc. Okay. So, let us load the data first, and since this is a CSV file, so we will use this command read.csv. Okay. Okay. So, it is giving me an error, probably because we are not in the right directory. Yeah. So, okay. So, we are in the right directory now, and let us try loading this. Yes. So, let us look at the data again, just to remind you how the data looks like. We have seen this before. So, we have expression data for 103 genes, and the first, so the column names are the gene names. So, you can see this ATP6V1G2, this is the column name, this is a gene name. Similarly, we have 100 such genes, right, and the last column is the label. Okay. So, this label refers to the cancer type. Okay. And we can also see this information in this labels file. This is also a CSV file. So, just read it as a CSV, and we can see what this file contains. We have seen this before. The first column is the cancer type, that is given here. Then we have abbreviation for this cancer type and the labels 0, 1, 2 and so on, right. So, these labels are the present in the data, right. So, instead of all these abbreviations or cancer type, just these labels are given. Okay. So, what we can do is we can see this, right, and what are the labels. So, we have labels 0 to 30. So, which means we have 31 cancer type data in this file, about 10,000 samples, and we can run this command. We have seen this before in dplyr package as well, right. What we are looking at is how many samples do we have per cancer type, per label, right. So, this is origin data, right, and we are familiar with this sign from this dplyr package. So, first is group by label, right. So, identify like which samples have the same label, and then count, right, these labels, and this is what we get at the output is label 0, we have 173 samples, n is 173, label 179 samples, and so on. We have a total of 10,000 samples, about roughly about 10,000 samples. Okay. So, this is the data, and as before what we do is like we choose only a small number of samples here. Okay. Again, this is just to demonstrate how this works. Of course, I will encourage you to run this with the full data set that we have, and that is something that could be very interesting, but it will take a bit of time, right. So, that is why we are taking only a small number of samples. We are taking 50 samples from 5 cancer types, label 0, 1, 2, 3 and 4, and we are also taking only 20 genes, right. So, we are familiar with these commands now. From the original data, we are choosing these rows, right, 1 to 50, then 174 to 2 to 3, right. This is something that we can see from the data, right, where we have seen the count data for every level, and then we are taking C1 to 20, these first 20 genes, and we are saying, right, take the label column as well, right. This is what we want, and we can see this again with head data. As you can see, we have again these genes, only 20 genes now because we have chosen 20, and then we have the label column. We can also look at the dimension, right. So, we have 250 samples, 5 cancer types, 50 samples each, and we have 21 columns because 20 genes plus the label. Okay. So, once we have this, now what I will do is I will kind of simply replace the 0, 1 with the cancer type, okay, just for our easy understanding, right, what we are looking at. So, this is cancer type LAML and so on, right. So, we know this from the label files, right, this data labels command here, we know this what are these, what 0 corresponds to LAML and so on. Okay, so this is done now, and what we are going to do next is we are going to divide the data into training data and test data, right. So this is something again we are familiar with, right, and you have seen this before, might have done this in different way, but here what we will do is we will now split the data, right. So what is happening is split initials, like this is the initial split data. So fraction would be 80%, so that is why it is 0.8 and based on the levels, right. So for every level we should have 80% of the data, right. So otherwise what will happen is we will take maybe all the samples from one cancer type and will not have any sample from other cancer type, right, that kind of thing. So which we do not want, right. What we want is take 80% of each cancer type in the training data and 20% of each cancer type into the testing data, right. So that is why we have this strata equals to level. Okay, and let us see. Okay, so we have training, testing, right, and the total. So it's saying 200 would be in training, testing 50 samples, and total is 250 samples. Okay, and then now we will split the data. So it's a very simple notation, as you can see: split training, split testing, and we can look into the train data, right, and we have the labels and we have the test data. Okay, and again we have the labels. Okay, so let us see if we have the training data. Yeah, so as you will probably notice, right, for training data we have this all cancer types, right. So 5 cancer types we have them. Okay, you can also look into this machine train, right. We have 200 samples and 21 columns, 20 genes plus the label. All right, so now we have generated this training data and test data. What we will need to do is we now need to build the model. Okay, and for doing that we will use this command called multi-norm regression. Okay, so because we are doing multi-class classification, right, we have multiple classes. As you have seen, we are working with 5 classes here, right. So since we are doing 5 class classification, multi-class classification, we will have to do multinomial regression, right. If we want to do logistic regression, we will simply say logistic regression, right. So we have this command logistic regression. So let us run this multinom regression, and here are some of the parameters we have: mixture equals to double 1, penalty double 1. This is actually talking about whether you should run at penalty, etc., right. Set engine using GLMnet, set mode classification, right, because we are doing a classification problem here, and this is the description or the formula of the model, right. So the formula is predict level based on whatever features we have, right. So based on expression data for 20 genes, predict the level, okay, whether it is a BLCA type cancer or whether it is a LGG type of cancer, that is what we want to predict from here. Okay, so with that, what we are going to do, let us run this now. Okay, and the data should be training, right, because we will feed the data on training data, right, feed the model on training data, and then okay, so let us see. Okay, so again we have 5 levels. This is what LPL means, right, and this is the feed, etc. What we can see now is the tidy model, right, and here are some estimates for this. Okay, so as you can see, this is what is saying, right, the coefficients for each of the features, right. So if you kind of remember, right, here we are estimating this probability, right, but as a function of this features, like as a linear function of these features, for which we have to estimate these weights or coefficients and the bias, right. So that is why we also have again intercept as well as the coefficients for each of these genes here. Okay. Now these coefficients will change for depending on the class, right. So this is what we will see. right. If you want to predict this ACC, these features will have different coefficients. If you want to predict the class LAML, these features will have different coefficients, right. So, that is why the coefficient values, if you remember, we wrote them as Wij, right. So, j is the 1 to k. So, the j actually varies, right, based on the class, and this is why the features, the coefficients for each of these features will be different. Okay, so, we have built this multinomial regression model, and now what we want to do is, now we have to predict based on the test data, right. So, as we have seen, we have built this model on the training data. So, data is trained. So, which means the whatever we are fitting here, whatever model we are building is based on training data. And now, what we are going to do is we are going to predict, right, on the test data, right. Again, we have seen this command predict. We are going to use this model that we have built. New data is test, type is class, right. So, we want to predict classes of these data points, and let us see what we have here. Okay. And this is what the predicted class will be. So, again we have 50 test data points, and we have 50 predictions, and what we have to see whether these predictions are accurate or not, right. So, now what we are going to do is we are going to look at class probabilities, and we are going to look at confusion matrix, and then maybe you can look into something called precision and recall, right. So, how precise the estimates are and how well this classification is done. Okay, so, this is what class probabilities we are calculating, and then followed by that we will calculate the accuracy and predicted probability, right. So, predict model new data test type is equal to probability, right. Earlier it was type was class, right. Now, type is probability. So, what it is doing is predicting for each sample whether it will belong to ACC or BLCA or BRCA or LAML or LGG, right. So, you can see these probability values for each of these classes. So, for each sample. So, rows are samples, these are the test data points, test samples, and columns are the classes. As you can see, these are the probability values, and for first few samples that you can see here, as you can see, the probability is highest for LAML, right. So, as we have discussed, right, since this probability is the highest, so the model will predict, okay, they belong to the LAML class, okay. And so, this is what logistic regression or multinomial regression will do, and what we are going to do now is we will combine this, right, results, select label, and we will bind columns, okay, predicted class and predicted probability. Okay. So, what is happening is we can see now again. Okay. So, maybe we can look into the head. Okay. So, this is the actual level of the data, right. This is the actual class of this data. This is our ground truth. This is the predicted class from the multinomial regression, and then we have the probabilities that we have just seen, right, the predicted probabilities. So, just in case we want to look up the probabilities later on. Okay. From this, what we can do now, we can calculate the accuracy value, okay, and the accuracy is simply taking the results. The actual truth is the given by these labels, right, and estimate or prediction is given by this dot predicted underscore class, right. So, you can see these columns. So, this is in the data, this is the actual label is the ground truth, and this is the predicted class, and we will see how well do they match. Okay. And the accuracy, as you can see, this is saying this is accuracy, this is a multi-class classification, and estimate is 0.92. Okay. So, this is 92% accuracy. So, that is a pretty good accuracy already, okay, to start with. What we want to do, right, we still have some error, right, in this prediction, and what we want to look at is something called confusion matrix. Okay. What it will show, the confusion matrix, is something like this kind of structure. So, the rows are the predictions from the model, and the columns are the ground truth. Okay. And what ground truth is telling, okay, this sample belongs to this class, and the prediction is saying, okay, this sample belongs to this class, right. So, what we will expect, right, for a good model is that most of the data points will lie on the diagonal, right, because this is what this means is that the model is predicting the correct level. Okay. So, in case of a good model where you want high accuracy, you want all the numbers to be along this diagonal, because what it will mean the prediction matches the ground truth. The numbers that are not along the diagonal, right, these are referring to misclassification. So, how do you read this? So, ground truth is this one sample actually belongs to this class SCC, but is predicted by the model as BRCA. Okay. So, this is a mistake that the model is making. And similarly, you have two samples here that are actually BLCA type of cancer, but the model is predicting as BRCA. For rest of the cases, for example, we have zeros, so which means there is no misclassification here. Here we have again one sample, this means one sample that actually belongs to LGG class is being predicted as SCC. Right. So, this is kind of referring to the model performance, right. Why is it important to look into this confusion matrix? Because sometimes accuracy might be high because the sample because the model is able to predict 4 classes very accurately, but is a very bad predictor for one class. Okay. So, this is something we can rule out by looking at this confusion matrix. We can also calculate something called accuracy and precision and recall, right. We have already calculated accuracy. These are again some other measures that are related to something to the accuracy, but they are looking at different aspects of the model. Okay. So, we will not talk about that, but as you can see the numbers are very high, 0.934 and 0.92. So, that is actually good. We want them to be close to 1, right. So, 1 is close to 100% accuracy or 100% precision. So, these are above 90%. So, that is actually is good for our prediction, means this model works very well here. Okay. So, this is something we have seen. The other model that we want to implement is on the noise data set. Now, one of the things I just want to point out, right, okay, this model gives us very good prediction, but we need to be actually have arrived at this stage by doing a very careful feature selection by applying something like the PCA and so on, where we have chosen actually those top 100 genes that will actually give us a good model. Right. So, what you see, like the implementation of the model is not so difficult, right. In R we can implement this model once we have the data. It is actually the feature selection and arriving at the right set of features, that is what matters. Okay. So, we will take another example of the noise data set in the next class, and we will see like whether the accuracy is high in that case, and then maybe if it is not high, can we improve that by doing penalized logistic regression or stepwise logistic regression. So, that will be for the next class. So, let us now move on to the presentation again. So, just to summarize what we have seen in this class. So, as I said, right, logistic regression is used for binary and multinomial classifications. We have talked very briefly about the theory, but what we have seen in the hands-on is that we could actually use multinomial regression to classify cancer types from gene expression data, only expression of 20 genes. So, that is a very powerful model, but as I told you, right, the feature selection is a very important part there. We have arrived at those 20 or 100 genes that actually will give us the best model. And then the other hands-on that we will be doing is classification of genes into noise classes in the next class. Thank you.